Hey guys, so in this video we're going to be talking about our adaptive immunity. So this is kind of like the second part of our immune system tour. So this is going to be the more specific immune system that we have. So let's go ahead and get started. So in the first video we talked about the innate immunity, which is where it's less specific, whereas the adaptive immunity is definitely a lot more specific, a lot more quicker. So the adaptive and innate immunity happens whenever you're born, whereas adaptive immunity does take time to develop after we've been exposed to an antigen or a pathogen. So the key components of adaptive immunity is that it is very complex, and then also it is slow developing, like I said, and then it does create a memory response in the sense of that if you were exposed to a flu whenever you were 3, and then you get exposed again whenever you were 6, you'll have a more robust and more specific immune response as compared to when you were 3. So you'll have a better immune response whenever you were 6. And we'll go into why that is. And then there are two components that you can kind of break the adaptive immunity into, and that's humoral and cell mediated. And we're going to go dive into both of these. So with the humoral, this has to do with our antibodies. So our antibodies, they are produced by what is called a plasma cell. And we'll go into how do we actually get antibodies and the different types of antibodies we have. And then also it's constantly circulating our bloodstream, and they will recognize foreign structures, and those are called antigens. So foreign structures can be anything, but for us, we're primarily going to focus on like pathogens. And then we have cell mediated. This has to do with our T cells, and T cells, they will also recognize antigens, and then from that they will stimulate B cells, or depending on the type of T cell, it will actually destroy the infected host cell if they come in contact with a compromised sick cell. So with that, let's do some basic vocab before we get started. So we have antigen. So antigen is derived from the term antibody generating. So this describes something that can elicit an immune response. So anything and everything that can cause an immune response is considered an antigen. For us, like I said, we're going to primarily focus on the infection part of antigens and everything. And then we have something called an epitope. So an epitope is the specific binding site of the antigen. So this is where an antigen will come in contact with a T cell, let's say, and then it comes in contact at the epitope. And then we have something called a heptin. So this is a small molecule, but whenever it attaches to a larger protein, it can act as an antigen. I'm not too concerned about heptins, but I really just want you to know the definition at that point, because for y'all, in my opinion, it's not as relevant. But definitely, if you're going to know any 3 of these, please know antigen, because they're going to be a reoccurring theme in this lecture. Okay, so with that, this is kind of just a basic diagram that kind of shows the big picture of the adaptive immunity. So this shows like the humoral and the cell mediated. We're going to go through what part is the humoral and what part is the cell mediated throughout this lecture. So at that, I'm just going to leave it. If you want to, you're more than welcome to pause the video and look at this. We're going to start, I believe, with the humoral side, and then we're going to go into the cell mediated side. So the humoral side has to do with all the antibodies and everything. We're going to go into different structures. Cell mediated has to do with the T cells and everything, and presentation of an antigen to a cell so we can get better. Okay, so with that, we're going to do one more definition slide, and then we're going to jump into antibodies. So the first definition that I want you to know is antigenicity. Sorry guys, can't say it. But this is a measurement of how well does an antigen actually elicit our immune response. So a lot of times if an antigen is protein based, we're going to have a very, very robust immune response to it. Whereas compared to if the antigen is just made up of nucleic acids and lipids, such as fats, it's not going to be a very strong immune response. That's all I want you to know at that point. but the reason why proteins elicit a very stronger immune response is because their structure is very unique, unique, and then that's mainly the big reason is because proteins are very, very unique as compared to lipids. Lipids are very bland in their structure as compared to proteins. That's pretty much it that I want you to know for that first one. And then the second one is threshold dose. So we kind of seen this before with antibiotic drugs. So it's kind of the same idea where you want to have the perfect amount of antigen presentation in order to generate the optimal response. So the idea is that you don't want too much antigen, because if you have too much antigen that is causing a response, then what can happen is that our B cells can become tolerant. So kind of like a like an alcoholic, where they become tolerant to the alcohol and they need more and more and more in order to elicit that same feeling. That's kind of the same idea with antigen presentation. And then also, if you have too low of a dose, then there might not be any activation. So you may not. So in the example of the alcoholic, it has to do with then they don't feel the buzz that they want, the high. So it's kind of the same idea with our antigens. And then we have the last one is immunological specificity. So this is where antibodies, they are generated for one sole antigen ever. So this is how we get actually a very, very good immune response, because our antibodies are very, very specific. And we'll go into how do we actually generate so many antibodies for different types of infections. So like there's one antibody specifically for like smallpox, there's one antibody specifically for the flu H1N1, then there's another antibody for the flu, let's say H2N2. So antibodies are very specific, and they will only respond to the specific antigen that they were made for. So with that, let's go ahead and talk about what is an antibody. So antibodies, they are this Y-shaped structure. I do want you to know this structure. I do want you to know the components of an antibody. So I'm going to highlight the big ones. So this yellow region is the variable region, meaning genetically, whenever we make our antibodies, that is the region that is actually going to change, and that is actually what makes antibodies very unique. So every antibody is going to have a different variable region, and that's how we get our diversity. And then the green area is the constant area. So this, every antibody is going to have the same constant area, always and always. And then they are held together by something called a disulfide bond, which are these S's in between the chains. And then these chains, so the ones on the outside, the short ones, those are called light chains. And then the ones in the center, these are called heavy chains. So I definitely want you to know that. And then also, one last thing about the variable region. So whenever you have the variable region of the light chain and the heavy chain, it creates something called an antigen binding site. So this is where your antibody will actually bind to the antigen, will bind to, like, let's say a pathogen. That's where the binding will occur. So definitely remember that, okay, guys? And then some vocab for antibody diversity. There are 3 ways that you can kind of break it up. There's isotype. So this is the type of antibodies that are found in all individuals in a related species. So meaning that you can find that antibody in multiple types of species that are closely related. And then an allotype is an antibody that is found only in one species, such as it's only found in humans, only found in dogs, or only found in cats, and so on. And then we have idiotypes. Idiotypes are antibodies that are specific to a person. So such as, like, my antibodies are going to be different from my parents, from my sister, from the husband. Like, that is specific to you. Okay? Okie dokie. So with that, let's go into the different types of antibodies. There are 5 that I do want you to know. So there is IgG. So if you see anything that's called Ig, that is called immunoglobulin. That can be used interchangeably with antibodies, okay? So we have IgG, IgA, IgM, IgD, and IgE. So I definitely want you to know those 5. And then From those 5, I want you to know what is their structure, what is their purpose, and where are they found. That's kind of the big components that I want you to know. And with that, I'm actually just going to go to the next slide because I feel that the next slides, they go over it a lot better as compared to this table. So with that, let's start with IgGs. So IgG, they're a monomer, meaning that there's only one Y structure. As you see, there's only one. These guys are the most abundant, so you'll find them in a lot of places. So you'll find them in your blood, in your lymphatic system, and then your intestines. Also, the fact that it's so small, it's able to cross the placental barrier, which is very good because that means that if it can cross the placental barrier, it can also aid in protecting the fetus. And then also their whole purpose of IgGs is to go around phagocytosing and then also neutralizing environments that may have toxins in them. So they're very, very simple in a way, but they are extremely important. It says IgGs. And then next we have IgAs. So IgAs are a little different. They are a dimer, so they have 2 Y structures. They're held together by something called a J chain. These guys, they're not as abundant as IgGs, but they are fairly abundant compared to the other ones. And then these guys, they're specifically found in tears, breast milk, and saliva. Excuse me. And with that, they aid in protecting the mucosal surfaces from invading pathogens. So that's all I want you to know for IgAs. And then with that, we have IgMs. So IgMs, they are the biggest of them all. They are a pentamer, so they have 5 Y structures. They're held together by that disulfide bond that we saw between the heavy and the light chains. They are found in the blood, lymph nodes, and they are also found on B cells. And what they do is that they essentially go around sequestering, so they go around collecting microbes. And then with that, they kind of preoccupy the microbes, and then they also aid in presenting them. And then with the fact that they are the first, with the fact that they sequester microbes, they are generally the first antibody to respond to an infection. Okie dokies. Okay, with that, we're going to go to IgGs. So IgGs, they're like IgGs where they are monomers, but they are not as abundant. They, just like IgMs, they're also found in the blood, in the lymph nodes, and the B cells. Whenever they are on the B cells, what they do is that they will actually initiate an immune response. Okay, so that's pretty much all I want you to know about IgGs. And then we have IgEs, which is the last one. He's also a monomer. He's the one that is the least abundant as compared to the other ones. So as we went through them, their abundancies or their occurrences also decreased, okay guys? IgEs, they're on mast cells, basophils, and they also circulate the blood. These guys are associated with allergic reactions, as well as they will aid in lysine parasitic infections. So cool for IgEs. Okay, so with that, we're going to go into the primary and secondary immune responses. So primary B cell responses, so this is all for the humoral side of our immune system. So the primary response is where an antigen will actually bind to a B cell, and then from that, what will happen is that once it's bound, the B cell is activated. And then once the B cell is activated, meaning that he's no longer like in a dormant state, he'll start proliferating, meaning that he'll make more of himself, and differentiating, so meaning that he will essentially choose like a career path for himself. And then from that, we can make plasma cells, and from plasma cells, we can make antibodies. And then after that, we have a secondary response, and this is where B cells are already activated, And some of them are kept as memory cells, so meaning that they are exactly what they sound like. They remember the infection, or they remember the antigen, I should say. And then whenever the memory cells encounter that antigen again, what will happen is that he will recognize it and he'll be like, this is what we did last time. We need to do this again. So it's a lot quicker, whereas rather than going through the whole filing process, essentially, like trying to remember what did we do, these memory cells already remember and they don't need to phone a friend to figure out what happened. So I hope that makes sense. But remember, primary response is the binding of the antigen to the B cell. Just remember that, okay? And then from that, you get memory cells. That's the big picture of this slide. Okie dokie. So, in class, we didn't talk about this, and we're not going to talk about this. I'm not too concerned about this slide, but I do want to talk very, very briefly about clonal expansion. So clonal expansion, this only happens in B cells. Oh, excuse me. So clonal expansion is how can I say? So this is whenever B cells, they will increase the number of themselves essentially, and then in order to make more of them, so they want to make more of self, and they will proliferate, and then they'll also differentiate. So whenever they make the clones of themselves, they can expand into either a T cell or a B cell. And then, but that has to happen after the first exposure to an antigen or to a germ, to a bacteria or something. And then with that, you can actually create a better protection against that same antigen later on. And then because of the fact that we have these new cells, the response is quicker, it is more robust, and this is why once you've had a illness, you don't get sick when you're exposed to it the next time around as severely, or actually you may not even get sick at all because of the fact that your body recognizes it and takes care of it. So you actually may have been exposed to the flu, but then your B cells and everything took care of it before it actually caused a problem. So that's all I want you to know, essentially. I do want you to know that it does, if we do keep our memory cells, they are stored in the lymphatic system, so, and the spleen. That's pretty much all I want you to know. But here is a figure of it explaining it. So this is like the original exposure. So remember, antigen and B cell binding are specific. Everything is specific, guys. Like, you can't just bind to everything, okay? So we see that the C4, this C4 cell, B cell, it binds to the antigen. So the antigen are the red dots. And then based on that binding, what will happen is that he'll be like, okay, so I'm good for this pathogen. I'm good for this antigen. I need to make more of me so we can make memory cells. So if we are ever exposed again, I can go in and attack more quickly so we can protect our host. And then also, so some of the clonal expansions will become memory cells, and other ones will become plasma cells, and those plasma cells will make antibodies. So that's kind of what I discussed about in the previous slide. And that's all I want you to know about clonal expansion. So with that, let's go ahead and move on. So let's talk about receptors. Oh, did we skip this in class? Yes, we did. Okay, so we did skip this in class, but I believe I mentioned just the 2 different ways of activating a B cell. So with that, I just want you to know. Oh, I think I skipped that too. Yeah, we're gonna skip that. Just kidding, guys. Okay, so with that, let's talk about how do we actually get antibodies to be very diverse. So, like I said, the diversity of an antibody happens in the variable region. So, this happens through just rearrangement of genes over time. Also, we have random mutations that just happen whenever we're copying them. So, mutations happen all the time. And then also, whenever we rearrange the genes, sometimes we'll make different codons. And then, based on different codons, we get different proteins. So that's how we get diversity um, in our antibodies, and that's pretty much all I want you to know for that. Okay, so with that, now we're going to transition into T cells. So this has to do with the cell-mediated portion of the adaptive immunity. So with that, let's talk about the different types of T cells real quick. I do want you to know the different types of T cells. So T cells, you can lump them into two groups. So we have T cells that have CD4 on their cell surface, or we have T cells that have CD8 on our cell surface. And then based on that, we'll determine their function. So we have T helper cells, so T helper cell 1, 2, 17, and Treg. And then we have cytotoxic, which are TCs. So all T cells, except for cytotoxic T cell, will have CD4 receptors. Okay? And then based on that, we'll also determine a certain protein. I believe I mentioned it before, the MHC protein. So the only cell that recognizes the MHC1 protein are cytotoxic T cells. Okay? And then based on what T cell you have determines its function. So everybody so you can kind of think of all your cells like ants. They all have a specific job and a specific destiny. So if you are a scout ant, you are only there for scouting. If you are a queen bee, then you're only there for breeding. So you can think of your cells that way. Like they have a specific job, specific function. They are made for a reason, and that's it. If they don't fulfill their reason, then they are discarded. So that's kind of a morbid way of thinking about it, but it's pretty accurate. So everybody has a function, and they all have a cell fate. Okay, so with that, we're going to go a little bit into like how do we actually activate our T cells. So some T cells, they're kind of dormant, kind of like B cells, and they need help to be activated. And then also we're going to go into T cell education. So how do they actually learn to become a Th1 or a Th2 and so on and so forth. So with that, let's go into T cell activation real quick. So T cell activation, in order for a T cell to know what he's supposed to do, he has to be activated by an antigen presenting cell. So the antigen presenting cell, for the most part, might be like a macrophage or even like a dendritic cell, like we talked about in the innate immune system. So these are guys that literally keep antigens or like pathogens on their cell surface, and then the T cells, they come around and they're just like, oh, I got you. Now I know what I'm supposed to do with my life, essentially. So whenever the antigen is presented, it's presented through a specific protein, and those are the MHC proteins. So there are two different types of MHC proteins. I just want you to know the differences between them, okay? So MHC1, they're found on all cell surfaces, all nucleated cell surfaces, sorry guys. So if that cell has a nucleus, he has an MHC1. And then these are specifically for presenting intracellular pathogens, meaning inside the cell. And then we have the MHC2 proteins. These are only found specifically on dendrites, macrophages, and B cells. So, and then they present extracellular, so meaning that they'll present it outside of the cell. Okay, so that's pretty much all I want you to know about the differences between MHC1s and MHC2s. Now with that, let's go ahead and talk about T cell education. So how do T cells become educated? Meaning, how do they determine what are they going to do with their life? So all T cell education happens in the thymus. So how, like, we know that the thymus is an organ very, very important to our immune system. So thymus is where all the T cells are learning where do they need, um, what do they need to do, and that's through two selections, and that's a positive selection and a negative selection. So the positive selection, this selection will ensure that the MHC restriction by testing the ability of the MHC1s and the MHC2 is distinguished between self and non-self. So, meaning that the T cells, these guys, they will weakly recognize self, and at the end, a cell will be allowed to live. So, in order for a cell to pass the positive selection process, the cell must be able to bind only to MHC molecules. And then if the cell binds to a non-self molecule instead of an MHC protein, it will fail the process and it will be murdered, essentially. So, what it is, I believe we already kind of talked about this in the other lecture, but so if a T cell binds to an MHC, the cell will live. Now, if I lost my train of thought. And then if a T cell doesn't bind to self, it will destroy you. I hope that didn't confuse y'all. But so if there is binding, if there is strong binding between the MHC protein, the cell will die. Now, if there's not strong binding, cell will live. Okay, so kind of like as we see in this figure, so we have a T cell precursor, and what he's doing is that he's going around and he's binding to MHCs and MHC1s and 2s, and as we see, it bound to an MHC2 and it was allowed to live. But if it goes through the negative process of cell binding, meaning that it was the negative selection, then what will happen is that it will die. So that X means that the cell died. So just know the differences between that. So that was, the first one was positive selection. So let's talk about negative selection. So negative selection, what it does is that it will bind to CD4s or CD8s. So as we see right here, so CD4 or CD8s, they're color coded. So CD8s are the kind of greenish, and then CD4s are like the teal blues. So what will happen is that binding will occur, and then what will happen is that when the T cell binds to a self MHC protein, so meaning that it's on us, that is presenting a foreign molecule, so that means that it has a foreign antigen on it, what will happen is that if the binding occurs, then if binding occurs through the CD4 and CD8, the self MHC, so our MHC molecule, is not presenting an antigen, or it's not presenting a self, so meaning that it's not telling the T cell that, hey, like, I'm one of you. What will happen is that it will fail the negative selection process and it'll be eliminated. So the fact that it didn't register it didn't tell the CD4, like, hey, like, I'm on your side, it will undergo apoptosis. So if it doesn't communicate correctly, then it's going to undergo cell death. And then without this selection process, we actually get a lot of autoimmune diseases, so that would actually be more common. But now the fact that we have this self from non-self recognition, we don't have as much as autoimmune diseases. Okay, so with that we have Tregs. So Tregs, these guys, they express CD4, strictly CD4s. They do express also other CD receptors, but I'm more concerned about y'all knowing CD4s. But Treg cells, what they do is that they aid in recognizing self from non-self. And that's pretty much all I want you to know. And then the fact that they help in recognizing self from non-self, so meaning recognizing our cells from bad cells. And bad cells can include like cancer cells too. It doesn't have to just be a pathogen. But with Tregs, they aid in reducing autoimmune diseases. So they aid in the fact that we're not always attacking our cells. We're only attacking the bad guys. Okay. So more on T cell activation. So with T cell activation, it is very, very specific. Like I said, everything is specific, guys. I don't think I talked too much about this in class, but I just want you to know that with T cells being activated, there will be some kind of signaling cascade that will result in something. So whether that be cell proliferation, so making more cells, or making cytokines, like if we have a TLR, like kind of like the TLR cascade, same thing. Just know that whenever T cells are activated through their receptor, there is a signaling cascade, and from that there will be some kind of response to it. But I know I did talk about this T helper cells, like how do they actually become activated by CD4. So T helper cells, they actually don't directly kill cells. The only actual T cells that does are the cytotoxics. But what the T helper cells do actually is that they help cytotoxic T cells as well as macrophages aid in killing infected cells, or they can also stimulate B cells. So if they're stimulating B cells, that means that they're stimulating antibody production. But then once a T helper cell becomes activated, they become activated by interacting with a antigen presenting cell. So that's the only way that they can become activated, okay? I'm not too concerned about y'all knowing the switching between CD40 and CD40L, but I do just want you to know that depending on what T helper cells, how do they actually activate, what's the purpose of them. So the whole purpose of T helper cells is to aid in activation of cytotoxic T cells as well as macrophages to aid in attacking affected cell, and then as well as stimulating B cells to produce antibodies. That's all I want you to know for activating T helper cells, okay? But I do want you to know specifically the 3 steps for cytotoxic T cell activation, okay? So there are 3 main steps, and they have to occur in order. So the first step, what happens is that a cytotoxic T cell will bind to an antigen presented on an MHC1 protein, okay? So it is specific binding, okay? So it's not just binding to anything. I hope y'all have finally got the clue that everything is specific. So a cytotoxic T cell will bind to an antigen presented on an MHC1. From that, the CD8 that is located on the cytotoxic T cell will come over and bind also to the MHC1 on the other cell, okay? And then from that, interleukin 2, so IL-2, is produced by a T helper cell, and then from there it has to come in and bind, okay? So this is a very much more in detail process, but if you can understand these 3 steps and then recognize a figure or even draw it out if necessary, if y'all like drawing, then that is beautiful. Okay? Okie dokie. And if you have any more questions about that, I can definitely send supplementals video for cytotoxic T cells. Okie dokie. So just a few more slides, guys. But once a cytotoxic T cell is activated, what it can do now is it can actually kill the target cell that it bound to. So it can kill the cell that it's now bound to by inducing apoptosis. So what it'll do is that a signal will be transmitted and it'll be like, you need to die, and then the cell will undergo cell death. But there is kind of a caveat with cytotoxic T cells. So since cytotoxic T cells, they bind specifically to MHC1, it can technically kill all cells that have MHC1. So that's kind of just a caveat of cytotoxic cells. But they're extremely important because of the fact that they can go around inducing apoptosis and the fact that they have high affinity for all cells. So that's a good thing. Okay, so 2 more slides, guys. I just want to mention something real quick about superantigens. I just want you to know what happens when a superantigen binds and where does it bind. So a superantigen, he's a molecule. You don't need to know specifically like what group of molecule or anything, but he's a protein. But what he does is that he binds outside that MHC complex that we've been talking about so much. And then if he binds on the right side of the MHC complex, MHC2 complex, let me clarify, MHC2 complex, it's not the one. What will happen is that there will be a giant release of cytokines. So cytokines will just come spilling out and then spilling out into the environment. Okay, so we have normal binding. So this is like normal binding between this T cell receptor and the MHC protein, whereas over here it shows the superantigen binding. So the fact that the superantigen binds outside of this complex, once it's bound, cytokines will be released. Okay, that's all I wanted to mention about superantigens. I'm not too concerned about it. It's kind of just more like a side note. Okay, now with that, one more thing. How do microbes actually evade our adaptive immunity? So there are some viruses that can actually downregulate MHC. So the fact that they're downregulating MHC, cytotoxic T cells can't bind, and then also it kind of hides them. And then also, or what they can do, they can actually utilize decoy MHC receptors. So essentially what they're doing is kind of like shooting blanks, or they're just like, look at these proteins over here, and don't look at the host cell with the actual true MHC proteins. So they're very, very clever in that sense. And then also what can happen is that there are pathogens that can alter the cell surface once they invade a host cell. So meaning that if it started off with a certain protein on it, if a pathogen invades it, it will change that protein on the cell surface. And if the protein is changed on the cell surface, then binding can occur. And if binding can occur, then our body's not going to recognize that we're being invaded. Also, there are pathogens that can induce cell death of T cells. So if it can fight off our T cells, then we're not going to have a very robust immune response, and our enemies are going to win, which is the pathogen. And then also, sometimes like how we mentioned that whenever a T cell receptor is bound to, you have a signaling cascade. So sometimes that signaling cascade can be interrupted, and that is where pro-inflammatory cytokines are blocked. So if that signaling cascade is disrupted and the cell doesn't realize like, oh, I need to put out inflammation cytokines, then our body's not going to realize we're under attack, because actually pro-inflammatory cytokines are good to a degree. I'm not going to go into detail as to where this become an issue, but you do want some kind of inflammation when you have an infection. Okay, so with that, that wraps up our lecture. I know we covered a lot of stuff, guys. If you have any questions, definitely don't hesitate to reach out. Let me know if y'all need any extra supplemental information. I could definitely post some videos that do a lot more visual aid than this video did. If not, I will see y'all in class, and enjoy your weekend.